first thing you need to know is that we found exceptionally well-preserved DNA in an abscess associated with this 800-year-old skeleton from Troy. The DNA within these abscesses turned out to be calcified nodules, which we originally thought were from tuberculosis because the nodule was found at the base of the ribs. But it turns out that they aren't. They actually turn out to be probably related to a maternal placental abscess from a female. This is a 30-year-old woman who died during pregnancy-related issues. In collaboration with Caitlin Pepperell at the University of Wisconsin-Madison, we sequenced the complete genomes of two interesting bacterial species from this placental abscess, Gardnerella vaginalis and Staphylococcus saprophyticus, both commonly found today in urinary tract infections. This represents some of the first archaeological and certainly the first genomic evidence of maternal health from Byzantine time periods. So we know relatively little about the burden of maternal health in the past, except if you fast forward in time and look at the historical record, sort of in the last from the 1600 forward. So we know very little about what deep time, the early Middle Ages really looked like. I think it's important to study the past to understand human health today. We know very little about the genetics and genomics of major infection. We're starting to understand that from major epidemics such as the Black Death or the Plague of Justinian. But the reality is that most people succumbed or had high mortality rates due to commensal bacteria, due to an imbalance with nature and a common infection that would consume them. In this particular case, what we see are two very interesting bacteria that are commonly associated with females today that in unsuspecting cases can reach up into the placenta and cause massive infection. In the case of Gardnerella vaginalis, the diversity in the sequence that we see in the ancient strain looks very similar to the diversity we see today. So that means that diversity has been maintained in that species for 800 years. In the case of Saprophyticus staph, we see actually a very unique strain 800 years ago, and that has shifted over time in a sort of coevolution with humans, and that is most closely related, the ancient one, to bovine or cattle strains. And so what we probably assume is that this was some sort of jump, perhaps, from bovine strains into females at some point in the past. We still live today, like we did in the past, in a delicate balance with our environmental microbial milieu. That is to say that we are constituents of both bacteria and human cells, and we have bacterial species that are living in symbiosis as commensals with us. And understanding how that balance can be disrupted, such as in this case with a maternal infection, is important in understanding basically morbidity and mortality, both in the past and in the present, and today into the future.